Greetings, future food safety officers. Bill Nye here. Today we're diving into something incredibly small but hugely important: cells. Now you might be thinking, science guy, I'm here to keep food safe, not to look through a microscope. But consider this. Everything that can make food go bad or even make people sick is made of cells. From the bacteria that spoil milk to the yeast that makes bread rise, it all comes down to these tiny building blocks of life. Understanding the two basic types of cells, prokaryotic and eukaryotic, is like having a secret decoder ring for food safety. It helps you understand why we have certain rules, like cooking meat to a specific temperature or washing our hands so thoroughly. Knowing the difference between these cell types is fundamental to your job. It explains why some troublemakers like E. coli bacteria are so tough and can multiply so quickly in a hamburger patty. It also explains why other organisms, like molds, behave differently, growing fuzzy patches on old bread. When you understand the basic biology of these microscopic critters, you're not just following a checklist, you're becoming a scientific detective. You can predict how contaminants might behave, why certain cleaning agents work better than others, and how to stop a potential outbreak before it even starts. This knowledge empowers you to make smarter, more effective decisions on the job every single day. Think of it this way. A car mechanic needs to know the difference between a gasoline engine and a diesel engine. They both make a car go, but they work differently, need different fuel, and have different problems. It's the same with cells. Prokaryotic cells are like simple, fast go-karts. Eukaryotic cells are more like complex sedans with lots of extra parts. Both can be found in our food systems, some as helpers and some as hazards. Your mission, should you choose to accept it, is to know your players. This foundational knowledge will make you a more confident and competent guardian of public health. So, let's get started on our cellular adventure. We're going to break down these two types of cells in a simple, clear way. We'll look at what makes them tick, where you'll find them in the food world, and why it all matters for keeping people safe and healthy. By the end of this, you'll see the world of food safety on a whole new level, the cellular level. And trust me, it's a fascinating place. It's science. And science is the key to understanding the world. From the biggest stars in the sky to the tiniest germs on a cutting board. Let's go. Here, we can see a simple diagram comparing a prokaryotic cell and a more complex eukaryotic cell. Notice how the eukaryotic cell has a clearly defined nucleus, while the prokaryotic cell features a nucleoid region instead. This basic difference is at the heart of how these cells function and why they matter so much in food safety. First up, let's talk about the prokaryotes. The name itself gives us a clue. Pro means before and karyon means nucleus. So these are cells that came before the nucleus. They are the original old-school models of life on Earth. Prokaryotic cells are incredibly simple. They don't have a true nucleus to hold their genetic material or DNA. Instead, their DNA just floats around in a tangled-up region called the nucleoid. Think of it like a messy room where all the important papers are in a pile in the corner instead of being neatly filed away in a cabinet. This simplicity is their superpower. Because they are so simple, prokaryotes can reproduce very, very quickly. Under the right conditions, like a warm, moist kitchen counter, one bacterium can divide into two in as little as 20 minutes. Then those two become four become 8, and so on. This is called binary fission. Before you know it, you have millions of them. This is why leaving perishable food out for just a couple of hours can be so dangerous. The prokaryotic pathogens like Salmonella and Listeria are having a massive party and multiplying at an incredible rate. Their structure is basic: a cell membrane, cytoplasm, ribosomes to make proteins, and that nucleoid with the DNA. Most prokaryotes are single-celled organisms. The most famous examples in your line of work are bacteria. Bacteria are everywhere, and many are harmless or even helpful. We use bacteria to make yogurt and cheese, but the ones you'll be focused on are the pathogenic bacteria. These are the prokaryotes that cause foodborne illness. Common culprits include Escherichia coli, often found in undercooked ground beef, Salmonella, linked to raw eggs and poultry, and Clostridium botulinum, a dangerous character that can thrive in improperly canned foods. These tiny, simple cells are the primary reason we have rules about cooking temperatures and food storage. So what makes them so tough? Many prokaryotes have a strong cell wall outside their membrane, giving them structure and protection. Some can even form a tough dormant shell called an endospore. This allows them to survive extreme conditions like heat, drying, and chemical disinfectants, only to wake up and start multiplying again when conditions improve. Understanding this resilience is critical. It's why clean isn't just about what you can see. It's about eliminating these microscopic, simple but incredibly persistent prokaryotic cells from food preparation surfaces and equipment. They are the invisible enemies you are trained to defeat. Here we get a closer look at Salmonella bacteria, which have a simple rod-like shape. Even though they look pretty basic, these tiny organisms can cause big problems if they end up in our food. Their simplicity is what makes them so good at surviving and multiplying, and that's why it's so important to understand how they work. Now, let's move on to the other team, the eukaryotes. So, EU means true, and karyon, as we learned, means nucleus. So, these are the cells with a true nucleus. This is the single biggest difference. Unlike the messy room of the prokaryote, a eukaryotic cell has its DNA neatly organized and protected inside a membrane-bound nucleus. It's like having a secure filing cabinet for all the cell's important blueprints. This organization allows for a much higher level of complexity. All the plants, animals, fungi, and protists on Earth are made of eukaryotic cells. Yes, that includes us humans. Eukaryotic cells are not just more organized, they are also full of other specialized parts called organelles. Think of a cell as a tiny factory. The nucleus is the main office, but you also have mitochondria, which are the power plants generating energy. You have the endoplasmic reticulum and Golgi apparatus, which are like assembly lines and a shipping department, packaging and sending out proteins. You have lysosomes that act as the cleanup crew, breaking down waste. All these little organelles work together, making the eukaryotic cell a marvel of complex machinery. This complexity means they are generally much larger than prokaryotic cells. In the world of food, eukaryotes are everywhere. The apple you eat is made of plant cells, which are eukaryotic. The steak on your grill is made of animal cells, also eukaryotic. But for a food safety officer, you'll also be concerned with other eukaryotes, specifically fungi, like yeasts and molds, and some protists, like parasites. Molds that grow on bread, cheese, or fruit are eukaryotes. While some are used to make foods like blue cheese, others can produce harmful toxins called mycotoxins. Yeasts are single-celled fungi that can cause food spoilage, but are also essential for making bread and beer. Parasites are another group of eukaryotes to watch out for. These are organisms that live on or in a host. Parasites like Giardia lamblia, a protist found in contaminated water, or Trichinella spiralis, a roundworm found in undercooked pork, are made of eukaryotic cells. Because they are more complex than bacteria, the methods to control them can be different. Freezing, for example, is very effective at killing parasites in fish, while cooking to specific temperatures is key for meat. Understanding that you're dealing with a complex eukaryotic cell, not a simple prokaryote, helps you choose the right safety strategy. Here, we have a colorful diagram of an animal cell with its nucleus, mitochondria, and other organelles clearly labeled. It's a great way to visualize just how organized and complex eukaryotic cells really are. So, let's put it all together. Imagine you have two boxes. One is a small, simple cardboard box with one item inside. The other is a huge compartmentalized toolbox with many different tools, each in its own special slot. The simple box is our prokaryote, and the complex toolbox is our eukaryote. The most important difference, the one that defines everything else, is the nucleus. Prokaryotes do not have a nucleus. Their DNA floats in the cytoplasm. Eukaryotes have a true nucleus that houses their DNA, keeping it safe and sound. This is the number one must-know distinction between the two. This core difference leads to many others. Let's talk about size and structure. Prokaryotic cells are tiny and simple. They lack the complex, membrane-bound organelles that eukaryotic cells have. A bacterium is essentially a single open-plan room. A eukaryotic cell, on the other hand, is like a mansion with many different rooms, each with a specific job. This is why eukaryotic cells are typically much larger than prokaryotic cells. You could fit hundreds of bacteria inside a single human cell. This difference in structure affects how they live, eat, and reproduce. Next, consider how they reproduce. Prokaryotes use a simple process called binary fission. They just copy their DNA and split in two. It's fast, efficient, and allows for explosive population growth. Eukaryotes have a more complex process. Single-celled eukaryotes like yeast might bud, but multicellular organisms use more intricate processes like mitosis for growth and repair. This slower, more controlled reproduction is a hallmark of their complexity. For food safety, the rapid reproduction of prokaryotes is a major concern, as a small contamination can become a big problem very quickly. Finally, let's think about where we find them. Prokaryotes are always single-celled organisms like bacteria. Eukaryotes can be either single-celled, like yeast or amoebas, or multicellular, like plants, animals, and fungi, molds, for example. In your job, you'll be battling single-celled prokaryotes like Salmonella and E. coli that cause infections, and also dealing with eukaryotes like molds, which can produce toxins, and parasites, which can cause disease. Knowing which type of cell you're up against helps you understand the risk and the right way to control it. Simple cell, simple rules. Complex cell, different rules. Isn't that neat? Here, we see a side-by-side comparison table showing the key differences between prokaryotic and eukaryotic cells, like the presence or absence of a nucleus, the types of organelles, their size, how they reproduce, and some common examples. This quick overview really helps to cement those differences in your mind. Alright team, you've done great. You've explored the microscopic world that is so critical to your work. To make sure this all sticks, let's wrap up with the essential takeaways and a little quiz to test your new knowledge. Think of these as the most important tools in your new cellular toolkit. Mastering these concepts will make you a more effective and knowledgeable food safety officer. You're not just enforcing rules, you're applying scientific principles to protect public health. That's a big deal, and you are up to the challenge. The journey of learning never ends, but you've taken a giant leap forward today. Here are 10 key points to remember. 1. Prokaryotes are simple. Eukaryotes are complex. This is the main idea. 2. Prokaryotes have no nucleus. Their DNA floats in a region called the nucleoid. 3. Eukaryotes have a true nucleus that contains their DNA. 4. Bacteria are prokaryotes. These are your main targets for foodborne illness, like Salmonella and E. coli. 5. Molds, yeasts, and parasites are eukaryotes. They pose different risks like toxins and parasitic infections. 6. Prokaryotes reproduce very quickly by binary fission, which is why food safety time limits are so important. 7. Eukaryotic cells are larger and contain many specialized parts called organelles. 8. Some prokaryotes form endospores, making them very resistant to heat and cleaning. 9. Plants and animals are made of eukaryotic cells, meaning most of our food is eukaryotic. 10. Knowing the cell type helps you understand the risk. Bacteria cause infections, while molds can create toxins. Now, let's see what you've learned. Don't worry, it's an open book test. The goal is to reinforce these important ideas. Take a moment to answer these questions for yourself, and then check your answers below. Pop quiz. 1. What is the main difference between a prokaryotic and a eukaryotic cell? 2. Which type of cell are bacteria? 3. Where is the DNA found in a prokaryotic cell? 4. Name one foodborne illness caused by a prokaryote. 5. Molds and yeasts belong to which cell type? 6. What are the small specialized parts inside a eukaryotic cell called? 7. Which cell type is generally larger? 8. What is the rapid reproduction process in bacteria called? 9. Why is rapid reproduction in bacteria a food safety concern? 10. Name a food safety concern caused by a eukaryote. Answers: 1. The presence of a nucleus. Eukaryotes have one, prokaryotes do not. 2. Prokaryotic cells. 3. In the nucleoid region. 4. Salmonella, E. coli, Listeria, any of these are correct. 5. Eukaryotic cells. 6. Organelles. 7. Eukaryotic cells. 8. Binary fission. 9. A small number of bacteria can become a dangerously large number in a short time. 10. Mycotoxins from mold or parasites like Trichinella.